So our next speaker is Ullasa from IISER Thiruvananthapuram. And now we move to two talks on plants.
Good morning. Thank you for the opportunity to speak about my work, our lab's work. So today I'll be talking about plants. I'm not a plant biologist, but Professor Renee wanted me to talk about evolution in the wild, and she would rather have me talk about plants than my butterfly work. This is the first time I'm presenting this work. But I also want to point out, I'm going to talk about this group of plants, Impatiens. So Uma talked about birds, mammals, and Robin talked about birds. Very few endemic species in India. This genus, Impatiens, has 120 odd species. 90% of them are endemic. So this is in the Western Ghats, and 90% of them are endemic to the Western Ghats. So there's a lot of speciation happening in the Western Ghats. So this gives us a nice contrast with the previous talks. Right. So some of the work was done in collaboration with Dr. Deepak Barua in IISER Pune, but primarily this is a work done by two of my PhD students, Sumaya and Sneha. In a sense, I'm compiling all of their PhD work into a few slides here. One of the big questions in biology is why we have so many species. A one-word answer is evolution, or you could say speciation. Speciation is the process, the evolutionary process through which we get new species. Right? So we are broadly interested in speciation in our lab. We also work on other things like color pattern evolution, coevolution of butterflies with their host plants, and so on. But today I'll be talking about this work on Impatiens plants. Just so that we are all on the same page, a species can be defined as a set of individuals that can interbreed or potentially interbreed. So two species are different because the individuals of these species cannot interbreed with each other. For example, take a horse and a donkey. They can mate, but this gives you a mule. But the mule, this hybrid, is sterile. This cannot reproduce again. So therefore, the horse and donkey are distinct, even though they can produce hybrids. In a lot of cases, in fact, in most cases, hybrids are not even produced. So you don't normally get interspecific, that is cross-species hybrids. For example, these two birds here have different song, and this keeps them reproductively isolated. So speciation begins as divergences between populations. So we can think of an ancestral species here. So this, you can imagine this as having two populations or two sets of populations. They start diverging ecologically. So you start getting incipient species, which are incompletely reproductively isolated. Over the course of time, this could take perhaps millions of years or tens of thousands of years. Over the course of time, they eventually diverge enough such that they are now reproductively isolated. So this is what we call speciation. So once speciation is complete, sometimes you can get hybrid formation, that is hybridization. And so this hybrid has a mix of gene pools from the two parental species. And if this hybrid is fertile, in other words, it can reproduce with one of the parents, in this case, this parent. So what has happened is genes from this parent have gone to the hybrid, and from the hybrid into the other species. So this is what we call introgression. Right? So once speciation is complete, if you have a lot of hybridization and introgression, potentially the species can merge back, the two species which were distinct can now merge back into the same species potentially. Right? So in the initial stages of speciation, the probability of speciation is given by two opposing forces. One is differentiation. This is ecological divergence between the populations. And then you have gene flow. So these populations are still compatible with each other. So there is gene flow. So the divergence is counteracted by gene flow. For speciation to happen, the divergence needs to be higher. Right? And once speciation is complete, the probability of maintenance of species as distinct species is given by these two opposing forces. One is ecological divergence, or at least the divergence, if not increase, it should be maintained. And there's homogenization because of introgression. If you have too much homogenization through introgression, the species will not remain as distinct species. So in my talk, I'll talk about these two aspects. I'll talk about these two opposing forces in one set of projects, and I'll talk about the role of introgression in another project. So if you think in terms of geography, you can have multiple ways in which speciation proceeds, but one common, easy to think of way is what is called as allopatric speciation. You can think of an ancestral species which was distributed in a given area. A barrier comes up. This could be a physical barrier like a mountain range or a river. And this barrier reduces gene flow between these two populations. And eventually these two can diverge into new species. So this is allopatric speciation. For example, the Colorado River in North America, once it was formed, it created a barrier, and it created the Grand Canyon. Now you have populations that are isolated on either side of the river, and it's thought that this led to speciation of these squirrels. Now we have two squirrels. One is found only in the north, one only in the south, and this is an example of allopatric speciation. This, you can even date this like using techniques what Robin has presented. So this seems to have happened about 10,000 years ago. Right. In the Western Ghats, we have these sky areas that Robin very nicely presented. So what Robin talked about, they're primarily in the central and southern Western Ghats. In the northern Western Ghats, so this is in Maharashtra primarily, you have these kinds of habitats. So you have these flat-topped mountains. So these are very rocky. So these are rocky plateaus. And you have valleys in between them. So these are essentially sky islands, right? Now, on the top, it's a very, very harsh environment. In the dry season, very, very dry, very hot. Very few organisms can survive. During the monsoons, there's no soil to seep out, right? For water to seep through. It gets waterlogged. And again, it's a very harsh environment. So plants and animals that live there need to be able to deal with these two harsh environments, right? And very few plants and animals can survive on these plateaus. So many of them that survive, they tend to be specialists. And one of them is this plant, which I'll just present. So this plant is, if you go to one of these plateaus in Maharashtra near Satara, and almost the entire plateau is covered by a bed of these plants. Very spectacular. And this happens just after the monsoons. This plant flowers, fruits, and the seeds fall down. By that time, it's too dry. The seeds germinate the next year. Right? When you go closer, they look like this. And these are the flowers. So this is a rocky plateau specialist. It's found nowhere else. And what really caught my attention was this. So this is Khas Plateau, where it looks like this. 17 kilometers away, a different plateau here called Chalkewadi. The flowers look completely different, right? To me, they look like two different species. It's not just the size, the shape, the color. This is deep pink. This is whitish, right? This is Toshegar. This is intermediate, 8 kilometers from here, 15 from here. The flowers are intermediate, right? So keep in mind, this is not the kind of scale Robin works on. This is a very, very small geographic scale, right? So this is what caught my attention, and I wanted to see what's going on, right? So a simple question. The strong specialization to rocky plateaus, is this driving allopatric divergences, right? Potentially leading to incipient species in this system. Right? This is Impatiens loviae. So this is what we studied. So as a control, as a comparison, we included another impatient species, Impatiens oppositifolia. This is found on the rocky plateaus, but also in the valleys. So it has a continuous distribution. So this serves as a nice control, right? A simple hypothesis. There should be stronger allopatric divergences in the plateau specialist compared to that in the generalist, that is oppositifolia, right? So this was very hard work going to the plateaus. Some of these are remote. Reaching there itself is a task, right? Students have quantified a lot of different flower. This is flower traits, but they also quantified other traits. So most flower traits are positively correlated with each other, which means they vary in a correlated fashion. So it's not just one or two traits that differ between the plateaus, but there are multiple floral traits. So here we have a representation of the differentiation within loviae. The three colors represent three plateaus, and you can see that the plateaus cluster out quite well. But in oppositifolia, which is a generalist, there seems to be no differentiation, right? As we predicted. This can be represented in another form. So here you have multiple traits combined here. You can see that the three plateaus are very well differentiated. In this is Impatiens loviae, which is a specialist, but in the generalist, there's no differentiation at all. Now, why do we have this differentiation? Any phenotype you take, the phenotype, you can have the effect of the genotype, that is variation in, if you have variation in phenotype, that could be because of variation in the alleles or the gene composition, or it could be because of the effect of the environment. For example, plants, their height, the branching pattern and so on can depend on the light conditions. Here's another example. I digress now towards butterflies. So this butterfly species has green and brown pupae, the cocoons, right? And there's a tendency for green ones to be found on green leaves and brown ones to be found on soil, rock, and so on. So it seems like where the pupate matters, right? And we wanted to know whether this is phenotypic plasticity. We've done a lot of experiments. So in one experiment, we can take these larvae, the caterpillars which are about to pupate, put them in these containers, green and brown, and here we have data on the color of the pupae. So higher values indicate more green. So if we force them to pupate on brown containers, they tend to be more brown. On green containers, the pupae tend to be green, right? So this demonstrates that pupal color is phenotypically plastic, right? Now, we could do this because we had a hypothesis about green versus brown, but with the Impatiens loviae, you can't do it because we don't really know what may drive, what environmental factor may affect their phenotype. So what we did was something in a way opposite to this. We brought all of them to the lab, and we grew them in what is called the common garden experiment, right? So here we have this differentiation in the wild. So now we bring them to the lab, grow them in greenhouse conditions. Now we ask, are these differences also seen in the lab, or do you lose these differences? If it's phenotypic plasticity that's giving you this variation in the wild, under greenhouse conditions, this variation should be lost, right? But if the plateaus are genetically different, if they're genetically differentiated, the variation, the differences between flowers should be preserved in the greenhouse conditions as well, right? So these were the experiments. So we took seedlings, so this is before they flower, brought them to the lab, grew them in the lab. This is what we see. This is data from the wild. This is for oppositifolia. This is Loviae. Here you have data in the transplant experiments. You can see overall the patterns are preserved, right? The differences are also there. This suggests that the plateaus are, the populations in these plateaus are genetically differentiated, right? And we also did this experiment with seeds. We collect the seeds, store them for 1 year. In the next year, we germinate them and grow them in the lab, in the greenhouse. Again, you see the same differentiation. Absolutely no differentiation in oppositifolia. Right? So it seems like the plateaus are genetically differentiated. Keep in mind that the plateaus are so close to each other, just 15, 20 kilometers. Still, they're genetically divergent, right? Very strong divergences. Now, we did the same with vegetative traits. Vegetative traits are the non-flower traits, height, branching pattern, leaf size, and so on. So I won't go into the details, but I want to point out that the variation is inconsistent. You don't get the same patterns that you see in the flowers, right? So then we asked what's special about flowers, right? So this divergence seems to be primarily in flowers, this genetic differentiation. So are the pollinator communities different? Are they selecting for different flower colors, right? We took 2 plateaus. We looked at the pollinator visitation, all insects that come. So these are the raw data, so to say, and these are scaled in terms of proportion. You don't need to go into the detail, but all I can say is that, yes, there are differences between the plateaus we studied, but it does not seem like the differences are strong enough to explain the kind of variation that we saw in the greenhouse experiments and in the wild. Now, if you look at this, Chalkavadi and Toshegad are close to each other. Kas is far away, well, relatively more further away. But in terms of the morphology, Kas and Toshegad are very similar. Chalkavadi is the most divergent one. And this is also reflected in the principal components analysis. Chalkavadi clusters out, right? So there's something happening there. It's not just geographic. It seems like it's not just geographic distance. So we want to look at this. We did a more comprehensive survey. So we went to a lot of different plateaus and tried to identify all populations of Impatiens loviae, right? Not all plateaus have them. They're found only in some plateaus for some reason. And so we identified 7 plateaus which range across the entire north-south distribution. And again, you find the same pattern. In the specialist, the plateaus separate out quite well, whereas the generalist does not separate out. This again tells us that habitat specialization is driving divergences between populations. And these are further analysis which point out to the same pattern. Here you have, this is giving you some idea of morphological differentiation across plateaus. It's much stronger in Loviae compared to that in oppositifolia. We also looked at the population genetic structure using AFLP data. Again, you have the same pattern. In Loviae, you have strong differentiation. In oppositifolia, the control, the generalist, it's not so strong, right? And so now, we wanted to see what's the role of genetic drift versus natural selection, right? So genetic drift is simply differentiation between, so in this context, it is differentiation between populations by random chance, okay? Or you could have differentiation driven by natural selection, right? I'm simplifying this a bit, but it works for this presentation. Now, if you have genetic drift, what you expect is that they should be, as the differences, as the geographic distance between population increases, the genetic distance should also increase. Right? So that's a very straightforward prediction. So you should have a correlation. You could do either morphological or genetic distance. And the prediction is that this relationship relationship will be steeper in the specialist, and it's either absent or weak in the case of the, so it should be steeper for the specialist and weaker for the generalist, right? So morphological distance versus geographic distance, there's no trend at all. Genetic distance, yes, there is some signal for isolation by distance, suggesting that genetic drift does explain some of the variation that we see. But for morphological distance, for some reason, we don't have that, right? Then we looked at natural selection. I already talked about the pollinator data. The pollinators don't seem to be different enough to explain the patterns. And we looked at the effect of the climatic variables. And a simple, straightforward prediction is that populations with more similar climatic regimes should be those that have lesser genetic distance, right? For morphological distance, we looked at multiple climatic variables. There's no correlation with the geographic distance. But when we looked at genetic data, there are a few variables. For example, precipitation data can explain this, some of the genetic differences that we see across populations. To wrap this up, strong specialization seems to drive allopathic divergences across populations, and this can be explained both by genetic drift and environmental selection. Now, very quickly, I'll talk about the second part. This is about introgression. So, in this species, Impatiens balsamina, so this species is found as you drive along, it's found along the roads as a very dense set of individuals, very common, right? Now, there's one more species called Impatiens rosea. So Impatiens balsamina has a very long nectar spur. The nectar spur is where nectar is stored. So pollinators have to land here, and they have to insert the proboscis or the tongues to reach the nectar spurs. Therefore, the pollinators for this species are likely to be different from the pollinators for this species, which has a very small nectar spur, right? So, in fact, Darwin saw these flowers, an orchid which was presented to him, it was gifted to him, and he predicted there should be a moth which has a tongue, a proboscis, which will be able to take the nectar from this plant. So, but at that time, no moth was known which had a 30 centimeter long proboscis, but a moth was indeed discovered later on. Right. So here, this is the Deccan Plateau. So here you have the high altitude mountains, the rocky plateaus. You have the Konkan plain along the coast, and you have the Ghats, right? So this is where if you drive along one of the Ghats in the Maharashtra, you climb down or climb up this Ghat section, right? So the Ghats look something like this. So we found balsamina when we were surveying this for some other project. We found balsamina only along the Konkan, the coastal plain, rosea only at the tops, at least in the area where we were working. Interestingly, along the Ghats, we found hybrids which had intermediate sized nectar spurs. So this, we didn't expect this to be there, but we found this. And then we went up and down multiple times, and we found it very surprising. You start at the top, you find only balsamina, very, very abundant. So you find only rosea, very, very abundant. You drive through the Ghats, you suddenly see hybrids, only hybrids. You reach the plains, you see only balsamina, right? And what was even more surprising, this is at the top, you have only rosea. So here, this is where you have the Ghats. So we found the hybrids, just a few kilometers, very narrow, and then you have balsamina. Keep driving, again you get the hybrids. This is all along the lower elevation. A few kilometers away, again you get rosea, right? Very weird. In fact, in this river, this is Savitri River, on the northern side, you drive along the coast, you get rosea. After a little while, you get balsamina, again you get rosea. In the southern one, you have a different species. So we wanted to understand what's happening here. So first we wanted to confirm whether these are actual hybrids. So we did morphometric analysis. So it's not just a nectar spur, there are many other traits which we included. Yes, the hybrids cluster out between rosea and balsamina. We brought them back to the lab and did cross-pollination experiments. So you take pollen from one parental species and drop them into the other species and so on. And here you have again the same pattern. So the lab hybrids, the lab population, they're in squares. The wild ones are in circles. You can see again all of the lab hybrids cluster out with the wild hybrids, which tells that yes, there can be hybridization and you can replicate that in the lab. We wanted to understand why is there this pattern? How are the species separated out, right? So this is a climatic niche model. To summarize this very quickly, you can look at the color patterns and you can see that all of them have the same climate suitability. It's not that one is more suitable, so to say, for the higher altitude and lower altitude. Their climatic niche, the climatic niche is the same. So there is more or less complete overlap. That does not explain why do you have this geographic patterns in the wild, right? So I'll skip this part. And now I'll come to this more interesting. Why are the hybrids so narrow? Why is the hybrid zone so narrow? Why are the populations not mixing up entirely? Why? Right? That's a question we wanted to ask. Right? One possibility is that the hybrids have very low fitness. Right? You do get hybrids, but they have low fitness. They have low survival or reproductive fitness. Again, we did these analysis in the lab. You can take pollen from one, create hybrids, and so on. So we have a lot of data, but I will just breeze through them. So here we have one estimate of fitness. This is seeds per fruit. Here you have the self-pollination treatment, that is, so this is for Impatiens balsamina. You take pollen from balsamina and place it on balsamina. You have cross-pollination, which is where you get hybrids between two species, between balsamina and rosea and so on. So now, you see that there's no differentiation. The hybrids don't really have lower fitness. And again, here you have germination rate. This is seed germination percentage. There's no difference between the hybrids and other treatments. We looked at many different traits. Hybrids do just as well as the parents, at least under lab conditions, right? Many other things we have looked at. Pollinator visitation in the wild, we don't find much difference. Surprisingly, even the nectar spurs are so different. They have the same pollinator communities. These are at least the diurnal, the day active ones. We have not looked at the night pollination, right? Then we wanted to check whether there's a difference in adaptation. Balsamina may, for some reason, be better. It may have higher fitness in its native range compared to that in the foreign range, right? If you take balsamina and grow it in the rosea's range, what happens, right? So we did these experiments. We did these transplant experiments. We take plants from the hybrid zone, transplant them to the balsamina rosea zone. You take the rosea ones, transplant them to the balsamina zone, and so on. We have a lot of data, but I just present a couple of slides. Here, for example, you have survival. The red curves indicate survival in the foreign zone. The cyan ones are survival in the native zone. So you see here, for example, survival is a little better in the foreign zone. Here it's a little better in the native zone, and so on. So there are some differences, statistically significant differences, but there's no strong pattern. What it means is that they survive just as well. They have the same fitness, irrespective of where they grow. And the question is, why the hybrid zone so narrow? Why is it so narrow? Just a few kilometers. Why is it not getting mixed up, right? I don't have a good answer to this, but we have some data from the population genetic analysis. So we've done DD-RAT sequencing. What we've done is we've taken a transect. So this is one hybrid zone. So from Mulchi, this is rosea. This is the Mulchi hybrids. You have the balsamina. Then you have Dapoli hybrids and rosea, right? So the Mulchi rosea and, okay, this is not intended, but anyway, the Mulchi rosea is here. The Dapoli rosea is here. What can I do? No. Okay, the magic fingers worked for it, right? So they are the most distant, geographically distant, but they belong to the same species, right? Here is a table of pairwise FST values. FST is simply something that, I need to wrap up. FST is something that tells you how different the populations are genetically, right? So higher values indicate more genetic distance. I just want to point out that the highest genetic distance was between Mulchi rosea and Dapoli rosea. So the same species, two populations, they have the highest differentiation compared to everything else, right? Very, very strange. And then this is also reflected here. If each column here is one individual, and the color represents its ancestry, genetic, determined by the genetic composition, right? So the Dapoli rosea has a completely different color, which means its ancestry, it's a different genetic cluster, right? The Mulchi rosea is different. Then you have Balsamina, which is represented by pink and so on. Without going into much detail, it seems like I. rosea, the two populations are genetically the most distant, and we also seem to have some suggestion of introgression, and this is asymmetric. Introgression is more from Balsamina into rosea than the other way around. So what we think is that the hybrid zone is dynamic, right? It's recently formed, right? And the Balsamina may be invading into rosea. Balsamina may be pushing rosea, making it smaller and smaller. If you follow this over multiple decades, perhaps we'll see this change happening, right? Yes, thank you. I think I overshot by a minute. Yeah, thank you.
Just very quickly, you now have genetic data. Have you estimated genetic diversity in each of these, and do you find any evidence of bottlenecks? Could there have been bottleneck effects? Because that actually can completely obliterate the relationship between genes and geography.
Yeah. So it's a very good question. We've not looked at the bottlenecks. We do plan to do those. But one thing our genetic data do suggest is that there's very high inbreeding, right? So these are called impatiens, possibly because the fruits, they're not dispersed. The primary mode of dispersal is not through birds, but they burst open, and the seeds, they don't disperse too far. So maybe there's very high inbreeding, which keeps the hybrid zone very narrow. That's potentially maybe what's happening here. Yeah.
Impatiens is a very complex genus, and we know that. And I think that is the best model for this study of evolutionary studies. So I was thinking that when you took those morphological, when the genetic distance was very low in case of the morphological analysis in comparison to genetic distance. So that was very less. So could be, I think some of the parameters might be missed. Yeah. Say for example, the calyx spur and spurless calyx. So I think there is hope to really investigate deeper so that you can get that higher distance also.
Yeah. Certainly, we've looked at what traits that we could look at, but I must point out that there is a weak correlation between morphological distance and genetic distance. So there is a correlation. Plateaus that are more genetically distant are also morphologically more distant.
But state live is a good model, certainly. You have proved it. Thank you.
Thank you.